Okay, I hope I'm at the right conference, because I will actually speak about chickens. Huh? So I'm the first one. Huh? So what I will try to do is just give you some kind of an overview of a lot of studies that have been going on butyrate, endogenous and exogenous butyrate, and intestinal health in chickens. So normally I don't start with pictures of chicken meat and so on, but this time I do. Huh? Just to tell you that actually, well, what people are willing these days, of course, is, well, safe products. Consumers, they really want safe products, salmonella-free, campylobacter-free. They want to eat happy chickens. Animal welfare is becoming much more important. They do not want residues. They want less antibiotic usage in animals and so on. So there's a lot to do these days about sustainable food and production of animals. What you see here, for example, this is just a picture of chicken breast of the US market, and you can read on the label there, no antibiotics, humanely raised, cage-free, so on. Of course, cage-free because it's a broiler, but still, there's a lot of things going on sustainability. And you know, well, probably I've been listening to this kind of talks quite a lot. These are the 3 pillars of sustainability. Social sustainability, which is clearly about how people look into meat consumption. It's also about welfare, antimicrobial resistance, and so on. Then you have, of course, the environmental sustainability, and luckily chickens have a low carbon footprint at the end, but still. And of course, also, then you have the economical sustainability. And of course, this is getting more and more important. And actually, when you try to raise chickens, broiler chickens, for example, we, of course, go for optimal animal performance. And this is somehow sometimes conflicting with some of the other sustainability pillars. Because by producing animals with a very fast growth rate, you actually make them much more prone to diseases. And this leads to welfare issues and to antimicrobial usage and so on. So there's a little bit of a conflict sometimes there. So one of the key questions or challenges is, of course, how we can improve animal health, animal welfare, without affecting animal performance. We need fast-growing animals that are healthy as well. That's basically the main challenge for the future. Of course, well, the first thing is safe products. I'm just going to give you a few examples of how butyrate is actually being involved in salmonella control. These are actually some kind of older studies we have been publishing already. It's already close to 14 years ago. But still, just showing you something that, well, at least for the working on butyrate and chickens, this is all how it started. So at those days, in the early 2000s, there were a lot of issues, of course, with salmonella. It's still the case, but at that moment, there was a huge pandemic. And actually, salmonella is a bacterium that is residing in the ceca. It is always in the hindgut, and this is, of course, the place where fermentation of sugars is also occurring. And at a certain moment, we, we used coated products with different acids in vivo trials, formic acid, acetate, propionic, and butyrate. And we could see that if you deliver butyrate to the hindgut of the animals, to the ceca of the animals, that has quite an effect on salmonella colonization. The more butyrate, the higher the butyrate levels in the ceca, the less salmonella you had at that organ. And this is the place where salmonella is residing, so it's actually quite important. What you see here, this is just data from an infection trial, okay? We just inoculate animals individually with high doses of salmonella, and this is the titers in log colony forming units. So that means a control population with about 10,000 to 100,000 bacteria per gram. And if you add butyrate, you see that it goes down with about 100-fold to 1,000-fold in colonization level. So this is quite something. This is salmonella shedding, butyrate supplemented group and a control group. So actually, again, showing you the same effects. The reason why this is the case is because butyrate, as you know, it's produced by a lot of bacteria, but it's for pathogens, it's very often some kind of a signal to reduce virulence. We don't know actually how that really specifically is working, but some pathogens sense butyrate even at low concentrations, and then the pathogens reduce virulence, and it's a very specific action. If you just take a test tube and you have butyrate added to the test tube and Salmonella, you will see that only the virulence genes are actually downregulated and not of global metabolism. So it's not really antibacterial, but it switches down bacterial virulence. What you see here, Salmonella is an invasive pathogen, meaning it goes into epithelial cells and then just goes to the bloodstream. Okay, and this process of invasion is very critical. If Salmonella is not invading epithelial cells, it will not colonize. So it's a very important aspect of the pathogenesis, and that's what you can see here. If you have a normal Salmonella strain and you inoculate a bird, you see that this is the colonization pattern. Even after 30 days, you still have high concentrations of Salmonella. If you use a non-invasive strain knocked out in a certain virulence gene, you see that this is not colonizing anymore. I'm just telling you that this process of intestinal epithelial cell invasion is crucial for the pathogenesis and for colonization. And this is actually what butyrate is doing. It specifically downregulates virulence gene expression, and it specifically downregulates invasion gene expression. So that is how this system is working. You have similar effects on Campylobacter. You also have other pathogens that react by reducing virulence and toxin production, for example. Not going to focus too much on this, but this is actually a cell culture assay showing you that if you pre-treat Salmonella bacteria with propionate or butyrate, that you have much less bacteria inside the epithelial cells, and for acetate you have more or less the opposite effect. So it's all about ratios between fermentation acids. If you have a lot of acetate and almost no butyrate propionate, you will have Salmonella colonization going up. If you have a lot of propionate and butyrate, you will see that colonization is going down and also invasiveness is going down. So this is just a small story on how we started working on butyrate, because of course at that moment we just used, well, a lot of companies started using coated butyrate products, but of course a lot of bacteria as well produce butyrate, and we started gaining interest into the potential of butyrate and the potential of adding products to steer towards butyrate fermentation. So, as I just mentioned, also animal welfare, less antibiotics is of course a key issue for future poultry production. And in fact, this is not always easy, because of course that means that, well, you have a lot of intestinal pathologies and intestinal syndromes and diseases in these fast-growing animals. And if we want to work with less antibiotics, we will need to preserve or prevent diseases. And this is what I will be focusing on in the next part of the slides. Just how can we promote health in the animals? And of course, I will mainly go into detail on butyrate production. Here you see a lot of pictures on what I call production diseases, okay? Diseases that are really only occurring in animals that are produced in the way that broilers are being produced, okay? You will not have a lot of these issues in your backyard chicken, simply because they don't grow so fast. And what you see here, well, there are quite a lot. You see, for example, white striping, what is a myopathy due to hypoxia, too fast growth, and even the capillaries or blood vessels can't even reach all tissue anymore, so you get some cell damage. Also, pathologies such as femoral head necrosis or other chondronecrosis, osteomyelitis, and this is also something, skeletal disease, that have to do with fast growth, definitely. And if you isolate bacteria from this kind of lesions, these are gut bacteria that you isolate. You isolate E. coli, enterococci, and so on. So this is also something that has to do with a poor intestinal integrity. And then, of course, the real intestinal pathologies, coccidiosis, I will not focus to it too much today, Clostridium perfringens and necrotic enteritis, and a lot of other enteritis syndromes. But important is that most of the antibiotics used in poultry practice, in broiler practice, is to treat intestinal problems. 60 to 70% is for intestinal pathologies. Just focusing a bit on necrotic enteritis, you know, well, this is the typical picture you see, this focal ulcerations, sometimes a little bit more, and this is caused by Clostridium perfringens, okay? This is a toxin-producing bacterium that is suddenly massively multiplicating in the small intestinal tract, producing toxins and causing gut necrosis. And again, we have done a lot of studies on trying to figure out how we can use alternatives to antibiotics for this specific pathology. And actually also, if you use an experimental model in which you try to induce this type of lesions, you also see that coated butyrate products are actually very effective. What you see here, this is a control population in which about 50% of the animals develop necrosis. When you start adding butyrate at different concentrations, you actually see that numbers of animals that have necrosis in the gut are actually much lower. Okay, so as I just mentioned, necrotic enteritis is in fact, well, it's an massive multiplication of Clostridium perfringens in the small intestine. And this is in fact the most severe case of what we call dysbiosis. And dysbiosis is something that's often used and misused in the field, I think, because, well, if animals are performing not well, a lot of people say, okay, they have dysbacteriosis, whatever it may mean. But Clostridium perfringens is definitely a dysbiosis, because if you sample the gut of these animals, you only have Clostridium there. But more important, I think, is all these types of intestinal syndromes that are like affecting performance, which if you take samples of the intestine, you see some kind of inflammation. Okay, and in broilers, it's very often the case. It can be due to whatever mycotoxins, maybe sometimes a little bit of coccidiosis, a lot of viruses, adenoviruses, reoviruses, maybe diet composition, or whatever. But then what you see is quite often this congestion of blood vessels, so that points to inflammation. And what you see here is in fact just histology on an intestinal sample, and all the brown dots here, these are lymphocytes. These are lymphocytes, that immune cells that have been infiltrating the gut wall. So this is inflammation. Here you see the normal slender long villi. Here you see the thick, blunted and fused villi and all the inflammatory cells. And this is, if you take samples from different flocks, you see this kind of excess of inflammation quite often. A few months ago, we took a lot of samples in different broiler flocks in the field, and we just gave these samples to the pathologists, and they thought these were diseased animals, because this massive inflammation occurs very frequently in broiler flocks. Now, focusing a bit on this, enteritis, this is what I just said, this inflammation you will get, and the main issue there is epithelial permeability, because inflammation always means that there's some kind of a defect. This is the epithelial lining. You also see these villi. If you have some kind of defect there, that means that some antigens will cross that barrier, make contact with some receptors, and start an inflammatory cascade. So epithelial integrity is the key issue to avoid inflammation. And inflammation is linked to performance, definitely. It costs a lot of energy for these animals that they have to spend and cannot use to gain weight. Okay. So just, well, if you have damage to any, whatever the trigger is, it's a virus, it's a mycotoxin, it's a clostridial toxin, it's even subclinical coccidiosis, normally a host can cope with that, okay? If you have a little bit of gut damage or epithelial cell damage, normally it is repaired very fast. Normally it's not a problem at all, because, well, you have many different strategies to maintain this epithelial integrity. Cells can proliferate very fast. Even small gaps can be sealed by just cells that connect again. No problem at all if the damage is not too severe. Tight junctions are very important also in this regard. And also, of course, a lot of anti-inflammatory responses can be imparted. And if you look at this slide, you will see that what you see here, and this is something that was in the first talk of Hervé, I think, already in the beginning, there are indeed, if you look at increases of cell proliferation, of cell differentiation as well, anti-inflammatory responses, you know that butyrate is actually doing this. So butyrate seems to be a very important molecule also in maintaining intestinal health, because it is very strong in maintaining, well, in helping to maintain and restore epithelial integrity. I just give you just one, some data of an in vitro experiment just to show you actually what butyrate can do. This is just a cell culture system. This is a transwell system in which you actually grow epithelial cells just in growth medium, and you can measure electrical fluxes through these cells. If you have cell damage, the electric flux is much higher. That's basically what we do. And this, is just a very tightly sealed epithelial cell layer. This is just an in vitro system growing a cell culture with all the cells that really connect with each other using these tight junctions. As you can see here, okay? You can damage this cell layer by different means, even by adding toxins or whatever, but you can also just add some cytokines. So you damage this layer and you will see that you have reduced resistance, electrical resistance. So the permeability is actually increasing. That's what basically this is saying if we add these cytokines. But if you, at the same moment, also add butyrate, you see that actually this integrity is completely maintained. So this is one of the most powerful chemical molecules in this kind of in vitro model systems. So it's really very affecting in preventing this integrity losses, and that's something you can very easily show in vitro. This is actually published. This was a gut paper of a few years ago that we have been publishing this kind of work. The same with metabolites produced by a lot of these butyrate producers. You also have quite a lot of other metabolites in general that have this kind of effect. You can also measure, cytokine expression. So this is IL-8, which is really one of the first cytokines produced by epithelial cells to attract granulocytes, so immune cells. And actually, you can do exactly the same story here. This is the basal level of IL-8 secretion in normal epithelial cells. You can, again, induce it. Look at the scale. It's going up to 1,000 and whatever. But if you add butyrate in the same medium, you also see that it's not going up that high. So butyrate is strongly anti-inflammatory. That's what I want to point out here. It helps to maintain epithelial integrity and is very strong anti-inflammatory component. You can show that in vitro, but also in vivo. It's something that is very well documented. So butyrate is doing these three things. It also helps to, for cell differentiation as well. So this is, I think, quite important to note. This is, I already have mentioned this. I just want to explain a bit about, and it's good that Petra gave a lot of introduction already on microbiota, because it has been very well documented in mouse models and in humans what beneficial microbes are to some extent, of course, for chickens. And some people have raised the question already, are the microbes the same in chickens and in humans? Basically it is. You find more or less the same genera and species. Of course, it will be other strains, but you find very similar patterns there. And what we have been doing is just looking into inflammation models in chickens and then figuring out how the microbiota composition looks like, okay? And this is this is a model. This is just a model, okay? We have been using in which we just give some kind of poorly digestible diet. We give a bacterial cocktail after we have been depleting the microbiota, some coccidia, and then we just do sampling and microbiota composition analysis. It's just a model. You can use any kind of model. You can use a coccidiosis model, a diet-induced inflammation model, and so on. It's just an example, okay? But what you see, and this is what we want to achieve in this kind of models, is, of course, body weight depletion. The animals grow less, and also they have some lesions, coccidiosis lesions, but also some macroscopical, a gut wall appearance that doesn't look completely normal. If you look at histology in this kind of models, you see, of course, villus length decreases, and it's quite, it's 25% decrease in villus length, and you see a lot of immune cells that infiltrate the gut wall. You see that here. This is a gut wall that is of these challenge conditions that is really full of immune cells. So this is just an example of an inflammation model in broilers. And what I want to come to is, if you look at microbial composition here, in this kind of models, you see that actually also butyrate producers are often depleted. And it can be because of the inflammation, because the inflammation also yields a lot of, well, oxygen radicals and so on, that maybe also ensures that these strictly anaerobic bacteria are depleted somehow close to the epithelial cells, so in the mucus layer. Okay, so this is just, we have been mentioning Faecalibacterium already. We have a lot of data showing microbial composition shifts. I just pick out a few of them. What you see here is Faecalibacterium prausnitzii abundance, and actually if you plot a correlation, it's statistically not the best way to go here, but still, if you do that, you see that the abundance of this germ is going up when villus length is going up, and especially this one is important, that if you have a lot of inflammatory cell influx, if you have an inflamed gut, that the abundance of this germ is very, very low. And I just picked out Faecalibacterium because it's an important one. You can also do that for other ones. This is Butyrate. This is one that we have been isolating many years ago. You also see the same trend, okay? And you can actually do that with a lot of bacteria out of this Ruminacoccaceae family, also some of this Lachnospiraceae family, and these are all butyrate producers. And I fully agree with Petra when she says that you can't just look at Lachnospiraceae, Ruminacoccaceae, you have to pick out different genera and then just, because sometimes you don't see this kind of bacteria going up or down, but others take over and others are depleted. So it's really a bit, it's probably because the function is quite redundant between some of these bacteria. But anyway, so it seems to be the case that a lot of these butyrate producers are actually, when the abundance is low, you get an inflammatory cell influx in the gut wall. And this was what is already mentioned a few times. A lot of these bacteria are coming from these two families, and these two families contain a lot of butyrate producers, but also others. Not all of them are actually producing butyrate. But basically, well, if you look at chickens, if you look at swine, at humans, and so on, you find a lot of these germs. That's basically it. So the principal mechanisms of beneficial microbes versus opportunistic pathogens, that whole concept is just everywhere the same, also over all the different animal species. This is something you cannot read, but it doesn't matter. These are a lot of strains that we have been isolating from chicken ceca before. And actually also Petra Lewis was involved in some of these papers. But what I just want to point out here is that all the strains that you isolate from chickens, actually they fall into the same genera and families than the ones you isolate from humans. And some of them, you see here, we have been also isolating from chickens, also Faecalibacterium, Butyrate, and so on. And if you use these strains, this is chicken isolate of Butyrate. If you use these strains in rodent trials as a model for human inflammatory bowel disease, they are very, very active as well. So they're not only active in chickens, they're also very active in models for human disease. And also Faecalibacterium prausnitzii for sure. Actually, one of these strains we just has been out-licensed to a company for use in humans. So this is, and this is chicken strain. So, okay, just give you some data. Again, this is the necrotic enteritis model in which we use Clostridium to induce intestinal pathology. If you use this kind of strains as a feed additive, you see that numbers of animals that develop lesions is actually decreased. These are 4 independent trials. The blue is the control population. Here you have, for example, 55% of the animals that develop lesions, and you actually decrease it in 4 independent trials, and also it works with some other strains. And it's a bit weird maybe that you just administer one strain, and this will not be because this particular strain is colonizing to a very high level and is producing a lot of butyrate. That's not the case, but it also changes the whole microbial composition when you just add enough. So if you start sequencing microbiota of an animal in which one of these strains have been administered, you see that there's a global microbial shift. Because you have to imagine that in this kind of models, you really have a severe dysbiosis. You have a lack of butyrate producers, and by just administering them again, you change the global microbial composition again. And this is really, well, this is a little bit shown here. This is again a trial in which we have been administering this Butyrate bacteria at very high concentrations. And this is just a snapshot of some data coming from small intestinal microbial composition analysis using 16S sequencing. And this is a trial in which we have been using just poorly digestible diets. And what you can do if you administer this kind of bacteria in these models is lowering feed conversion, so digestion is much more efficient. And in this kind of models, diet-induced inflammation models, you of course see again also these issues with villus length, immune cell infiltration, and so on. And by just administering this bacterium, you see that, well, actually lactobacilli populations go up, but also Enterobacteriaceae populations go down. And this is, I think, quite important, because these Enterobacteriaceae, these contain E. coli, not Salmonella probably in this case, but E. coli-like organisms that are actually opportunistic pathogenic. And we often see, if we take samples from chickens in these inflammation models, that if we induce inflammation, Enterobacteriaceae go up, butyrate producers go down, and actually you can play with feed additives to try to restore this particular balance. That's basically the thought of all these studies. You can see that here. E. coli is going down, Enterococci going down, some hydrogen sulfide producers are going down, and so on. And butyrate producers go up. This is just a small snapshot of all these data. Now, coming to something very practical again, how to increase butyrate, and maybe not cecal, but intestinal. You know that there are a lot of different compounds available to do that. Coated butyrate, esterified butyrate, and so on and so on. You have quite a lot of options there. Prebiotics in principle, but there was one question already on enzymes. And also this is something that actually has an effect on microbial composition. I will just show some data in a few slides. This is a slide, well, in fact, this has already been shown by Petra. Just the slide on how these acids are produced. It's a bit simplistic. It's a simplified scheme just to show that, of course, mostly the fermentation acids are a consequence of polysaccharide breakdown, using many different bacterial species or genera that all generate some steps in this breakdown process. But if you look at this graph here, and if you think about, well, products or feed additives or alternatives to antibiotics, there's quite a lot in here. You have prebiotics, you have maybe the pure acids itself, maybe you can add some bacteria that produce some of these metabolites, and so on. And also, of course, you can start playing around with enzymes, because if you help in breaking this down, maybe you produce some kind of oligosaccharides that have this prebiotic potential. So basically, first thing, are enzymes. Just that we just, well, have a lot of papers in progress using xylanases and other enzymes and looking at the effect on butyrate production and microbial populations. And simply said, if you just take a test tube, you take a fecal or cecal slurry and just add bacteria, and give it some substrate, and you add the enzymes in the test tube, you will see that also in a lot of cases butyrate production in this test tube is going up. So it means that at least it can be of value to add enzymes to diet of animals. But it depends on the enzyme, of course, and on the what it is really cleaving off and what is the end product of this enzymatic reaction. And just show you some data of a paper we published 2 or 3 years ago. This is just, well, this is just showing you, of course, the arabinoxylan structure, and this is the breakdown of these arabinoxylans, so arabinoxylans from plant cell walls. If you degrade it using different enzymes, you end up with arabinoxylan oligosaccharides. And one of the questions we have been asking ourselves before is what would happen if you really administer this kind of prebiotic substances to broilers. And actually, if you start doing this, you will see that, well, this prebiotic approach, you will see that actually it stimulates gut health to some extent. And in this particular study here, we have been again using some kind of diet-induced inflammation model. This is the feed composition, and you will see that this is not normal. There's a lot of rye, there's also rapeseed and sunflower meal, just to induce some state of, well, digestibility issues, bacterial overgrowth, and inflammatory cascades going on in the small intestine. And then we just used xylan-oligosaccharides at a 0.5% concentration in the diet of these animals. What you get is this here. So actually, well, this is published by a colleague. This is Céline de Maarschalk 3 years ago. And actually what you see is that also here, feed conversion is going down if you administer this XOS. So this means that digestibility is improved. Also body weight is going up, but also villus length is going up. And this is all significant. This is at day 26, so about 3 to 4 weeks of age. And this is the age to which mostly in this kind of in vivo models, the animals are most vulnerable or susceptible to this, well, drops in performance. So this is, well, what you see here is an xylan-oligosaccharide induced increase in absorptive surface, longer villi, so more epithelial cells. And it's very nice that Petra already has commented on this particular enzyme. This is one of the key enzymes in microbial butyrate production, and you can somehow quantify a bit using quantitative PCR, using the generated primers. And if you do that with the samples from the animals that received xylan-oligosaccharides as a supplement, you see that, well, it's much more homogeneous, okay? So this is a quantification in log units of the gene that encodes this enzyme involved in butyrate production. It's like a marker of, well, the capacity or the potential of a microbiota to produce butyrate. It's simplified again. It's not fully this, but it's a good marker for that. What you see is that if you, in the control population, there's quite some variability. Some animals seem to have much less of this butyrate-producing community. Others seem to have a bit more. But if you add xylan-oligosaccharides, you seem to enhance this kind of population. Okay, that's basically what this slide is saying. And even within the same diet, you see that there's quite some variability. And this has already been mentioned in one of the previous talks. One man is not another man, but also one chicken is not the other chicken, although they get the same diet, okay? And they're in the same room. So even then you have this kind of variability here. And that's also something you have when you look at microbiota composition analysis. Even within the same population, on the same diet, in the same room, you have these differences. But adding this kind of substrates seem to induce the capacity of the microbiota to produce butyrate. And actually, these are just some data. You see that specific groups of lactobacilli are going up. This axis is not completely valid, but Lactobacillus crispatus, for example, in colon. In cecum, you see that some specific genera or species even of butyrate producers are going up. And actually, what we tend to think that is happening is that you get some kind of a mechanism in which you have these xylan-oligosaccharides that actually are used by some lactobacilli to produce lactate, and then this lactate is being consumed by butyrate producers to produce butyrate. So it's this whole concept of cross-feeding, and that's why you need this high diversity, okay? That's basically what this slide is telling you. And actually you can model that in vitro. So if you take this strain and you add xylan-oligosaccharides, it will produce lactate. If you take this one, it's a butyrate producer, and you add xylan-oligosaccharide in a test tube, nothing happens, okay? It needs the lactate that is used by these bacteria, that is produced by these bacteria to further convert it. So it's really a cascade of events that lead to butyrate. That's why maybe if you start with chickens that only live for 5 weeks and not have a fully developed microbiota and are under a lot of antimicrobial use, that maybe this full cascade of and this microbial variability and all these different groups are maybe not present, and maybe the microbial composition of the animal is not adult enough to have a complete breakdown of all these components, and that's why maybe we need to help them with enzymes. That could be a theory. Again, a slide on Salmonella, and this is just showing you that these xylan-oligosaccharides or even other stuff that is fermentable to butyrate if you use Salmonella infection models, actually that is also something that seems to help in reducing colonization by this pathogen. Maybe it's because of the butyrate that has been produced as well. Because what you see here, this is just, what you see here is actually if you take a cecal dropping or fecal sample, you add it in a test tube, very simple, very simplistic as well, and you just add one of these components at different concentrations in a test tube, you see that actually this, the bacteria are producing butyrate out of it. We don't see that with cellulose and resistant starch, for example, in the test tube, of course. But if you also have an in-feed addition of these xylan-oligosaccharides, so you do infection challenges with Salmonella, you also see that the colonization level in the ceca, so the place of fermentation towards butyrate, is going down again. So it seems to be like there's a general concept of having more butyrate production, less Enterobacteriaceae, less Salmonella, less inflammation, and so on. There seems to be a very good correlation there between all different pathologies or intestinal issues that are relevant for broader production. Just a final slide already. Just to mention to you that anyway, well, there are quite a lot of studies showing that butyrate is important. There are quite a lot of studies showing that microbiota that is producing butyrate are very important. But the way how to enhance butyrate production in the gut can be very variable. There are a lot of different methods that can be used to stimulate endogenous butyrate production. I think the problem we have a bit is, in contrast to what is going on in human medicine, it's a completely different thing. If we speak about health in chickens very often, or if veterinarians speak about health in chickens, it sometimes means that they perform less. And actually, I don't think that's the same as health. If they perform less, why wouldn't they be less healthy? Okay. I think that's a major difference between what we are doing in chickens and what people are doing in humans. They actually have very different definitions about what health is actually, what health is really. So maybe one final remark here. You also have quite a lot of other molecules. Butyrate is very well known, but I think in the near future there will be a lot of other metabolites being published, produced by a lot of different bacteria. That actually target maybe the same receptors as what butyrate is doing, or maybe other receptors that are also very relevant for maintaining intestinal health. So this is the last thing I wanted to say. So thank you.